Everyone. It's great to see you all here. We're just waiting for the connection online to kick in. Brett has joined us. Brett, thanks so much for joining.
No problem. Well, thanks for having me.
Yeah, do you have slides to share?
I do. I have put something together.
Great.
Yeah. Is there any timeline or anything like that?
No,
Or
we're going to kick off in just a moment. We're just waiting for the online live version to go. So we have people rolling in now. It's just hit 6:30. We'll welcome everyone in just a moment. So if you want to pop your slides up, you are very welcome to. Awesome. Okay, let's kick off. Thank you all for joining us this evening. My name is Jess Taylor. I'm the CEO of QENDO. We have some really awesome people here who are joining in live online via Facebook, Instagram, social media platforms, but then some people in the room here. So we do have the team who are monitoring the chat and also online socials. So please feel free to ask questions or anything. If we can't get to the questions, we will take them offline and work with Brett and the team to hopefully answer them. But Brett, it's really great to be here talking about this with you. I am just going to acknowledge that I'm on Quandamooka land out in the Redlands and also pay my respects to the incredible storytelling that the tribes have done for tens of thousands of years. And that's what we're here to do tonight is to share and to learn and to share stories about what's happening in the research space within endometriosis. And Brett, my goodness, it may not seem like a lot to the wider public and sector, but there's a lot going on, isn't there?
there is at the moment. It's a good time to be in research in endo. A lot to do, but a lot happening as well, which is great.
Yeah. Yeah. Would you agree that a couple of years ago it was pretty dire in terms of the potential career progression? What were your thoughts as a, you know,
Well, yeah, it's always been dire, not just a couple of years ago, even
yeah.
before that as well. We've got a few things happening now, which is great, but I still think we're still behind a lot on what a lot of other life-threatening diseases are as well.
Can you tell me what do you think's been the shift? Why do you think that we've seen such a push finally behind the research, and why do you think we're actually getting some runs on the board?
I think it's great actually that the push is coming from people who want it, who need it. There's no point us in the research saying, "Oh, look, this is a really good idea. This is what we've got to do." It's when patients and advocates for patients are out there going, "Look, we need something done about this." That's when people start to listen, and that has been such a powerful force, definitely in the last few years, but even earlier than that, it's taken, they say there's no such thing as an overnight success.
Mhm.
That's very much the same in this space here as well. Like there's people like yourself and others that have been working very hard over a long time. It may feel like there's something coming out of that now, but it's not just because of the last few years. It's because a lot of people have needed this to happen for a long time.
Yeah, we stand on their shoulders and we're here because we can keep lobbying because of them. Awesome. Brett, before we jump into it, can I learn a little bit about you? What's your coffee order?
Just a flat white, unfortunately, not very exciting, but
No.
I probably have a lot of those per day.
Particularly around grant time and grant submission time, I'm sure.
Yes, yes, for
Yes.
sure.
We're caffeinated on coffee. Are you a dog person or a cat person?
I'm a dog person. We just have a new puppy in the house at the moment, actually. So
Oh.
you might hear some noise at the door if they're getting bored and want to come in. It's
Oh.
yeah.
What did you get?
So we've got a sheepadoodle.
Oh
Yeah, a cross
wow.
between a
Oh.
sheepdog and a poodle.
Yeah. Wow. What an interesting breed. What a mix.
Yeah, we had a sheepdog recently, but unfortunately we lost him this year, earlier this year.
I'm sorry.
So yes, it was time for a new puppy, and they
Yes.
are a menace at the moment.
Yes. What's their name?
Nina is its name.
Oh, that's so cute. So cute. Tell me why, Endo's a pretty hard space to get into. It's a really, which we hopefully we'll get into. It's a really complex area. It's a fascinating area of research. What keeps you in this space? Why did you jump in the space of endo research?
Yeah, I got into it by chance, is the honest answer. There was an opportunity to take a role in a research group that was working on endo. I knew nothing about it at the time. But after that, there's just so much space. Every question you ask hasn't been answered yet. So there's so many things to do. I don't think I've ever not wanted to get out of it. I just haven't finished what we started yet. So we
Mm.
just keep working on it.
Yeah. What I love about what the Institute for Molecular Bioscience and what you do and what you've taken on from all of your mentors over the years is really around that fundamental research, right? We know that we don't know really much about what these lesions are, what they do in the body, or very much it's not considered widely or accepted widely. There's not really any set agreements. Can you tell us, and we'd be keen to jump in now to tell us what is happening in the research space, where are we at with things, what's on the horizon, and what can we get excited about?
Yeah. Well, I think just to answer that question, I think, that's completely correct, is something that holds us back on the research side of things, on the treatment and the management side of things, is we really don't understand what disease we're looking at. It's really just a bunch of stuff thrown in together and said, this is endo, and we're trying to work out what that is. And so we can't even work out from a research point of view what we're dealing with. It's always going to make it very hard to work out what the correct diagnosis or the correct treatment for that's going to be. So that's very much, I think, one of the main issues in the field is just sorting out what exactly we're dealing with and then working our way through all of that. If we can sort that out, we can start to get better treatments and better diagnosis as well.
I often say we're currently throwing spaghetti at the wall and seeing what sticks, and we really need to know what spaghetti and what wall to throw it at.
Yes. Yeah, yes.
Yes.
Yeah, I think that's exactly right. I mean, it's very hard to say something. So looking from a treatment point of view, it's very hard to say something doesn't work when it does work for some people, but it just doesn't work for other people. And so we need to know why it works for some people and why it doesn't work for other people. And only then can we start making progress and just, throwing out the baby with the bathwater and looking for new things constantly.
Hmm. Yeah, I think the tricky and again, I'd be keen to hear your thoughts on this. The tricky thing with fundamental research or that basic research, the lab stuff, it's not as sexy as some of the the more fancy treatment stuff, and it's tricky to get some airtime on what you're actually doing. So that's a point of what we're trying to do tonight is help the wider sector understand. Gosh, it's just so important if we don't get and understand this work that you're doing and that IMB are doing, this is the building blocks and the Lego pieces that we need to then build onto all of those other initiatives.
Yeah,
So
for sure.
thank you for being here. It's this has been a long time coming. I think you and I have both said we need to get the wider group and sector out across what's happening. And there's some really cool things on the horizon. So I'll hand over to you now to go through your slides, and we will encourage questions and thoughts and comments, and we'll stop at the end of the slides and go through them. Thanks so much, Brett.
Okay, sure. So just to check, you can see the slides okay? I don't need to do anything from this end.
Yeah, we can see them.
Yep. Okay, great. So, thanks very much for the introduction. And just as Jess said, I'm going to talk a little bit today about the research we do at IMB. Sorry, I'll just go back to that slide because it's a nice picture. We're out at St. Lucia on the UQ campus. It's a lovely campus, and we've got lots of different stuff going on there. We also, I should say, work very closely with the clinicians at the RBWH because they're an integral part of our team as well. It's kind of what Jess touched on at the start there is, you know, the research we do, I think, is very much integrated into the fact that we need to understand some of the basic biology of this to really move that through the pipeline to see what we're going to do to improve diagnosis and treatment. We know that endo is a chronic disease, so the research and management of it needs to be focused on such a thing. So we have projects that really revolve around understanding the basic pathogenesis of the disease. We have the Endometriosis Research Queensland study, which you may have heard of. We look a lot at the genetics and genomics of all that. We also have other projects where we're focusing on the diagnosis and looking for developing different in vitro models that can help with diagnosis, but also treatment, looking at ways we can personalize the treatment. Again, what we just talked about, endometriosis is such a heterogeneous disease. We can't just be using a one size fits all. We need to be starting to think about ways that we can design treatments for individuals. And along those lines is finally we get to the management side of things, and that's where again our clinical collaborators at the RBWH are really important. We're looking at developing new ways of doing clinical trials so we can actually move all of this basic research into places that get back to the clinicians and to the patients themselves. And so, I've tried to put that together a little bit in what I can show here as well. So within our research group at IMB, we try and collapse all of those different projects, I guess, into 5 different themes that cover a lot of that information. And that's really covering clinical integration. So I talked a bit about the RBWH team, and it's really important for us at the front end of this to be able to enroll patients through our endometriosis Queensland research study, but also at the back end to get that information that we're working with back to the patients as well. We do a lot of work looking at genetic impacts of endometriosis. We know that endometriosis is what you'd call a complex genetic disease. So there's a collection of small but critical genetic variants that can increase your susceptibility to the disease. We still don't know how they increase the susceptibility. We just know they're there. So we're looking in a lot of ways to study that further. We're looking at leveraging that data to develop molecular profiles as well. And this is really getting towards the heterogeneity of the disease and understanding how we can improve treatment options. And so that's another one of our themes, is improving those treatment options along with improving non-invasive diagnosis methods as well. And so just following on from that theme is what I'd like to show is that I think the real power in what we do is having this research group integrated across these different areas is we can actually start to get towards basic science into the clinics by integrating these different types of themes. And so I'll show a bit of data where it's a bit of a case study on a project that we've had looking at the impacts of a particular treatment for endometriosis known as Deanogest. I think in Australia here it's called Visan, and it's just been listed on the PBS scheme. And so this is a project we've been trying to understand exactly how that works and how we might be able to select patients that could actually utilize Deanogest, and what do we do for the patients that it doesn't work for as well. So in that sense, we sort of split these into these themes, and one of those first themes is really using the genomics level to understand what the impacts of this treatment are. And just fitting that into context, I think it's already been mentioned a couple of times that this endometriosis is such a variable disease, and it really sits at the heart of what we're trying to understand. We need to understand that better if we're going to understand the disease and if we're going to target treatments to different patients as well. We know that there's a variation in presentation. We know that no 2 people have exactly the same symptoms, and also along with that, we know that the treatment outcomes aren't going to be the same. And so we really see that this is a significant barrier to getting better treatments. When we talk about what are the current treatments for endometriosis, we know that it's a hormonal-based treatment, and this is really designed to suppress estrogen at a systemic level. And you can do it with lots of different ways. You can do it with combined oral contraceptive pills. You can do it with progestins or GnRH analogs. Even within those, there's many different variations of the types of preparations that exist within those as well. And we know that with all of that, there's a variation in the way that patients respond, a really significant variation. If we look at progestins in particular, we know that up to 34% of patients can be treated with these and still have pain remaining after treatment. What we don't know is what is the variability driving, what is the factors driving this variability in treatment response. And so we can even go down a further level to look at that and look at the progestins in particular. So these are probably the most commonly prescribed hormonal treatments for endometriosis. But even within those, there's a huge variation of the different types of drugs that patients can get for when they're talking about progestins. And essentially, progestin, the only unifying thing between all of those is the fact that they actually target progesterone receptor. So because of evolution, other steroid hormone receptors that have significant effects around the body can also be targeted by these types of drugs as well. And so um, what that means is these different progestins can actually have significant off-target effects for some of these drugs. And so if we were to look at just a couple of the more common ones, being Deanagist or Visan, as I mentioned, it's quite specific for progesterone receptor, has activity on AR, which is androgen receptor, but doesn't really target anything else. In contrast, something like medroxyprogesterone acetate, or I think is known as Provera, is a drug that also has a target for progesterone, but has other off-target effects, and particularly at things like the glucocorticoid or mineralocorticoid receptors. So we can see that there is all of these potential other effects that these drugs might have. And so this is an interest we started many years ago, actually. We started looking at what the effects these progestins could have, and do they only work systemically on estrogen, or do they also work on the endometriotic lesion themselves and target some of these receptors? And we published a series of papers on this quite a few years ago, but this was essentially all done in the lab, not directly with patient samples. And so we sort of set up this project that we wanted to get to know a better understanding of what Deanagist actually does. Can we identify markers within that might suggest which patients would benefit from treatment? And what do we do, and how do we select other treatments that might go to patients that don't respond? And so we set up a project to do that. And knowing the heterogeneity of endometriosis, we were very strict with the inclusion criteria that we were set for this. We wanted it to be only one type of lesion, particularly deeply infiltrating lesions, because these are the ones that have the most difficulty in hormonal treatments. We wanted to know exactly the dose of Deanagist, and we wanted to be clearly defined which patients had taken the drug and which patients responded and didn't respond to the drug. We also went through a very careful process that we know when we get tissue from patients, firstly, the tissue has to go to the pathology department to do the diagnosis. So from the research side of things, we sometimes get left with bits of tissue that we're not quite sure what it is. So we went through a very specific process of making sure we had endometriotic lesions in all of the things that we studied. And then finally, we did this technique, this sophisticated technique that's called single cell sequencing, which I'll explain a little bit more because it sounds a bit complicated. And so what that essentially means is we're able to take these endometriotic lesions and isolate every single individual cell, and then run these through these small little pipes called microfluidics, which are essentially just small tubes. And when we run the cells through, we could capture them in their own little oil droplet and then amplify the genes within each of those droplets. And by doing that, we could put a barcode on them, and we could tell exactly what genes are being expressed by exactly what cells are being expressed. And we can do that for the lesions from every individual patient. And that creates a really powerful data set for us for a number of different ways. The first thing it does, it allows us to identify exactly what cells exist in endometriotic lesions. So we know that endometriosis is defined by the presence of epithelial and stromal cells, but we know that there's a lot more to it than that. And so from this project, we're able to accurately map what cells exist in these endometriotic lesions. And just at a very high level, we identified these 5 different cell types, obviously the epithelial and stromal cells that identify the lesions. But we could also see the endothelial cells. These are cells that line blood vessels. And importantly, 2 sets of immune cells. These are called lymphocytes and myeloids. So essentially, they're immune cells that control the inflammation. And so one of the first things that came out of this study, just looking at this mapping, is we saw this massive amount of immune cells in these endometriotic lesions. They were the second most abundant cells, these lymphocytes. And that's even considering that we selected the lesions based on the presence of their epithelial cells. So we were seeing that these endometriotic lesions have this huge inflammatory component. And so these are the lymphocytes here. And then, taking this a step further, when we looked at the cells that came out of lesions from patients that were either treated with the anagist or not treated with the anagist, we could see a clear impact on the inflammation that occurs within these endometriotic lesions. And so this was really critical for us, as it was evidence for the first time that the anagist not only has this systemic suppression of estrogen, but it's also impacting directly the inflammation that occurs in endometriotic lesions. And now I can get a little bit into the basic science here, so I'll go through this slowly. I hope I don't lose people. But essentially, what we did then was, well, okay, is the anagist working directly on these lesions? Can we find proof of that? So we looked for the targets for the anagist, in particular progesterone receptor and androgen receptor. And we could see that they were expressed in some cells in these lesions, particularly in the stromal and epithelial cells. When we compared the gene expression in these cells, we saw that there was a really significant downregulation of an important set of genes. These genes are called CXCL genes or chemokine genes. And essentially what these genes can be thought of is the genes that lead to the infiltration of immune cells. So when these are secreted from the cells, they lead to an inflammatory reaction. And when these lesions were treated with the anagist, we had a significant suppression of these chemokines. And that's, we believe, a reason why that we were getting this reduced amount of infiltration of inflammation in these lesions. Now, we wanted to check if this was in fact occurring at the lesions itself. And so that's one thing that we looked at more closely. We separated these between cells that were progesterone positive and androgen positive versus cells that were progesterone negative and androgen receptor negative. And when we looked at the expression of these chemokines, both before and after treatment, we could see there was a significant downregulation of these chemokines in the progesterone and androgen receptor positive cells, but there wasn't this similar downregulation in the negative and progesterone receptor negative and the androgen receptor negative cells. So we were getting pretty good evidence that there was a direct interaction between these drugs on the endometriotic lesions themselves. One of the things about chemokines, they attract in immune cells because they're able to bind to their receptor, which is called CXCR4. So CXCR4 is the main receptor for these chemokines, and it exists within the lymphocytes and the myeloid cells. So these immune cells that exactly we're looking to bring in to the endometriotic lesions. And again, when we looked at the differences in gene expression between these cells, when the lymphocytes or when these lesions were treated with the anagist, the gene in the lymphocytes that was the most significantly downregulated was this receptor for these chemokines. So we were seeing this real interruption between the ability of lesions to attract their immune and their inflammation. But opposed to what we saw in the other cell types is that when we split these between progesterone receptor positive and progesterone receptor negative, we still saw a downregulation with the treated cells. So essentially what that means is the anagist was having this impact on these cells, but it wasn't through strictly just the progesterone and the androgen receptor. So altogether, what we're seeing here is a real interruption of the inflammatory component that occurs when patients are treated with the anagist. Now, that's fine if you're responding to the anagist, but what happens for these patients that aren't responding to the anagist? And as I said at the start, we had this we had this cohort that had both opportunities available. And so we looked at the amounts of cell types also within these lesions. And interestingly, we didn't see an impact on inflammation between the patients that responded and the patients that didn't respond. The amount of lymphocytes didn't really change. What we did see was a slight change in the structural cells, what we'd call these stromal cells. So just get past that. So, and also when we look at the differential genes that were expressed and the pathways that were affected, we're seeing a very strong impact on the amount of fibrosis that occurs in these stromal cells. And when we begin to look at the histopathological images of these, we could begin to clearly see that there was a lack of these epithelial type structure, and there was a much larger fibrotic structure around these cells. And so it's not that easy for non-experts, but what you could see here is these sort of blue cells around here, these larger cells of the epithelial component. They were easily observable in the responding patients. In contrast, for the non-responders, we saw this much more fibrotic type tissue, so much so that the technician that was doing this work was pretty much able to predict which patient would be a responder and a non-responder when she was going through this process just by looking at the images. And so to put all that together, what we had was this potential, looking at these endometriotic lesions, we were seeing this direct impact of some of these progestins, particularly deanogist, where there is a suppression of this immune response through what we call these chemokines. There was less immune cells coming in, and that was creating a less of an inflammatory environment. For the non-responders, however, it was these structural type cells that were causing this fibrosis that were making it more difficult for these patients to respond. Now, as I mentioned, we like to combine the different aspects of the lab here. And so now that we know that we have a signature for what these patients might have that don't respond, we wanted to see if we could do that to potentially predict new treatments for these patients that don't respond to endometriosis. One of the ways we can do that is to leverage some of the available databases that exist around the world. One of these in particular is called the Connectivity Map. So it's a database that was developed by the Broad Institute. And without getting too complicated, it essentially is a set of signatures that they've worked out. Every time you treat a cell, it's these sets of genes change. And so if you have a really high positive score, you will get an impact that looks exactly like what that drug would do. If you want to reverse that signature, you get a really negative score. And essentially, it suggests drugs that may be able to reverse the signature that you don't want to see. So we use the signatures we developed from these non-responding patients to see what had a really high negative score. And some of the stuff that came from that was really interesting for us is that firstly, we saw that different classes of drugs affects the different types of cells individually. We saw that the types of class of drugs for epithelial cells were different to those of lymphocytes, to those of stromal cells. But one of the things that did come up that was very interesting to us was the fact that glucocorticoid receptor agonists had very strong activity on both lymphocytes and stromal cells. So this is interesting to us because, as I mentioned at the start, there is already progestins that have activity for glucocorticoid receptors, things like medroxyprogesterone acetate. So there's the potential, if a patient isn't responding to one of these progestins, they could be triaged into a different progestin with a slightly different profile. We also saw that there was the potential to target novel pathways such as the HSB inhibitors in epithelial cells, or pathways called NF-kappa beta in these as well. And so these are just classes at the moment. These are classes of compounds. We wanted to look more specifically whether there might be drugs within these classes that could be potential novel treatments for patients that don't respond. And so splitting the glucocorticoids into the multiple different compounds, we could see, again, a really strong negative tau score for pretty much all of the different glucocorticoid drugs that were in this database. So it's a really good basis for us to go forward and think we might have potential to treat these patients with a different type of compound. We also wanted to look for novel compounds, stuff that isn't related to endometriosis treatment. So these are drugs that exist clinically, they're able to be used, but they've just never been indicated for endometriosis before. So we went through a database and selected some different ones for those as well. And so to go through these steps now, we've used some of the genomic information to understand how deanogist might be working. We've then looked at the signatures created to come up with potential new compounds. But then I guess the next step from that is to start testing some of these compounds. And so that's also what we're trying to get into the lab at the moment. And as I mentioned, there, well, firstly, there's lots of different ways to test drugs in the lab. There's lots of ways to model the disease in different in vitro settings of what we call cell culture. So, for example, we have the ability to grow these stromal cells, which appear to be targeted by the glucocorticoids, or we can grow the epithelial cells into more advanced models, which we call organoids. And as the name suggests, these organoids are essentially miniature versions of the organ, and they have a three-dimensional structure, and allows us to get closer and closer to the real situation to see if the drugs that we want to use are actually going to be working as well. And so we took some of these drugs that we identified and then tested them on cells that we'd taken from patients from our endometriosis research Queensland study. Starting with the glucocorticoid drugs, we took patient cells from 4 different patients, treated those patients with 4 different glucocorticoids, and then tested how well those cells survived at the end of those 4 days. And as you can see in the graphs here, the untreated cells survived quite well, but pretty much all of the different glucocorticoids we used had a very significant effect on dampening the stromal cells and reducing their ability to grow. So it was really starting to validate some of the in silico predictions that we were making. But there's limitations on this as well. So these are with 2D stromal cells. We don't know how well they recapitulate the environment and whether they're going to be great models for the future. What we can do as well, though, is use these organoids that we were talking about before. So this is a step further from this and allows us to really shape things in a 3 dimension. So organoids are grown over a period of a couple of weeks where we will set these up into what's called a hydrogel, a 3 dimensional structure. And we can really start to grow versions of these patients in the lab so we can start to determine whether or not individual patients might respond to the individual drugs that we have. And so you can see here, there's a stylized version of an of a uterus of how it would look like. And here's a fancy 3 dimensional reproduction of one of the organoids that we're growing in the lab. And so we're getting closer and closer to being able to develop models that we can actually test these drugs on. And taking that to the next step, we decided to take some of these drugs that we'd identified and see if they would actually work on the individual patient organoids as well. So you can see here, so basically the way we would do that is we would grow the organoids. We would use some bioinformatics to be able to identify each of those organoids and count the size of those. We could see in the vehicle controls, we don't have much death. They grow really well. In drugs that are designed to kill these cells, they die off. And some of these really strong drugs that we'd identified were also dying off. But we would also have the potential to modulate the concentrations and allow these organoids to continue to grow or to see more and more cell death. And essentially what that would lead us to is results like this. And so I think this is a really important slide. So I'll go through it quite slowly. What we've done here is we've grown these organoids from different patients. And so we have 4 of these different patients represented by patient 1, patient 2, patient 3, and patient 4. And we've exposed them to all of these drugs that we've predicted might work in endometriosis. I think one of the key things to take from this is that essentially most of the drugs work similar for each of the patients. So the darker the color represents the stronger the activity against these organoids. And as you go down these, what we call heat maps, there is a gradual lightening of color. So the drugs that seem to work similar for some patients are stronger at the top and weaker at the bottom. But within all of that, we can see that there's really individual differences for all of these patients. For example, patient 2 here had pretty good reaction to most drugs. Most of the drugs that we applied were quite successful with killing off their endometriotic organoids. But unfortunately for patient 1 here, they weren't able to have that same level of success in the types of drugs, and they were limited more to these more specific types of treatments. So I think using this sort of activity, we can really start to highlight the fact that we need to identify what drugs are going to work specifically for what patients. And by using the genetic information we collected earlier, we can start to triage which drugs might be useful to go through these sort of processes. And so going back to this schematic and putting it all together, what we see is we see that there are certain drugs, a lot of these sort of hormones that can work directly on endometriotic lesions by interrupting the inflammatory profile. But there's also going to be non-responders to that need novel compounds. And we can utilize this information to start targeting different parts of those lesions. We can get potentially glucocorticoid receptor agonists to target the stromal compartment, but some of these other drugs, such as HSP90 inhibitors or NF-kappa beta inhibitors, that can target these epithelial organoid components as well, giving us more opportunities for the way we might want to approach treatment. And so this is all very much done in the lab still. This is all basic stuff that is only lab-based and we can't translate to the clinic yet, but it has implications for the way we might want to approach them. So we know that progestins can have a direct effect, and we know that patients characterized by fibrotic lesions are less likely to have effects to these drugs. So it's worth considering when there's adjuvant treatment defined for different patients, that if we use histopathological investigation, we may be able to select the right types of treatments for these people and triage them towards different types of compounds. All of that, though, really relies on getting a measurement of what's considered fibrotic and how we would select these patients. So there's still, while we have some evidence of this, a huge amount of work to go in before we can move that to the next step. And just to briefly put that into context, the last thing I would like to mention is to go back to that concept of working quite closely with the clinicians at the RBWH. So we now need a conduit for getting some of this laboratory-based work into the clinics. And that is coming very much through the work of Dr. Akwazi Amoako, who is setting up different clinical trials at the RBWH. And when I say different clinical trials, I think it's really important to go through what we mean by that. So previously, currently, the way to do a randomized clinical trial is you select the patient cohort, you decide what the intervention is, and you split those patients in two. You give them the placebo or the current standard of care treatment, you give them the intervention, and then in 5 years' time or whatever it is, you do the statistics to determine whether or not those interventions are working. Now, it's a very successful way to do it. It works, but it's slow and it's very expensive. It also significantly limits the number of trials, and for a disease like endometriosis, it becomes a very difficult way to get these individualized treatments tested in a way that we need to be doing. And so new versions have been proposed for this. And these very much came around the time of COVID, where we didn't have time to wait 5 years to understand whether or not drugs were working. We needed to be able to bring drugs in more quickly. We needed a more efficient evaluation, and we needed a way to take drugs out that weren't working. So these adaptive platform trials were developed. And so this is what we're doing in collaboration with the RBWH, is to get these adaptive platform trials up and running where we can get some of these drugs into clinical trials much quicker. The adaptive platform trials creates the opportunity to be able to, well, they're very similar to what is currently being run for clinical trials, except they have extras on these. They allow for the updating of statistical data regularly and for decisions whether or not interventions should continue or not. And importantly, as I mentioned, they can add in additional interventions. And so that can be described pretty much here in this, where say you start with a standard clinical trial, but one of these interventions isn't working. We know that it doesn't work, so it's no point continuing with it. We can drop that. If we have an intervention that we think might work, that we've seen evidence in the lab that could be good, we can add those into the clinical trial, and we can get these things moving more and more regularly. And so that's the basic idea of it. And as I said, I mentioned that because it is a work in progress with the RBWH at the moment. We are working through getting the infrastructure up to develop this, and hopefully aiming to implement this sometime in 2026. So we can start getting some of this stuff we're seeing in the lab and we're developing with organoids into a conduit to be able to get this directly into clinical trials within the patients. And so just to sum that up, uh, there was a lot there, I guess, and I think I've seen a few questions come through. So endometriosis research at IMB, I think we link closely with the patients and clinicians' needs to really try and identify how we can get the best treatments to the best patients based on what the heterogeneity of the disease is. We use cutting edge technology, cell biology and genetics to be able to do that. But what we're really trying to do is pioneer a lab to patient approach. And we're currently developing the infrastructure and framework to be able to do that. And so with that, that's it from me. And just thank you for those who've contributed and also for the funders that have helped get our work done. So thank you very much.
Thank you, Brett. It's just so wonderful to hear, and we're a really proud collaborator on the adaptive platform. I'm leading the consumer activities there, and we have several expressions of interest for the consumer panel that we're bringing together. So if you're interested, please email us and I can send over the details. So just to be clear, before we jump in the questions, you are growing endometriosis lesions outside of the body and in models that isn't
Yes.
mice to try and track what we're doing. What's it doing, right?
Yes. I see. So again, for the way we focus on the disease, we need to focus it on an individual. And so the way to do that, and for very much thankful for the patients that contribute to the study through the RBWH, is they provide that tissue to us, and then we will grow that in the lab in a way that to try and replicate as close as possible. We're not exactly there yet. There's a lot of work to do to growing an endometriotic lesion in the lab. And I think only once we start doing that can we really understand what's causing that lesion and what drugs might work on that lesion. So yes, I'm a big fan of large, big studies, but we also have to focus studies on the individual as well.
Mm. It's so much work, and I really hope that people listening, it's so much information and it's so much work, and we can get really frustrated where we think, oh, what's happening? Nothing's happening. But if you can really understand and where Brett has taken through the process and the amount of people, resources, brain power that goes into plan, establish, manage the risks involved, the bloody ethics committees involved, to all of that, and then bringing people along the way, and then actually having tissue to test on, it is an enormous project. So there is stuff happening, as you can see. It just takes time.
And I think what you said at the start, just all the question about why are we starting to make progress, it's every bit of that um, that process, all the patients, the clinicians, the basic researchers, the geneticists, the bioinformaticians, they all have to have a role in all this. And they're not the only, I'm sure I've missed other people out of that as well. But it's all of them pulling in the same direction to get somewhere. And, I think that is where we're starting to get to.
Whenever I present workshops, whenever I, train researchers or consumers, they always say to me, yeah, but, basic research, fundamental research, how does consumers get involved? And I think it's a really amazing example of the role of someone who's willing to share their tissue. And someone has said, and asked the question if they are de-identified tissues. Which of course they are, and
Yes.
they do have special tracking IDs that the appropriate people can access. But from your end, Brett, they're de-identified, and that's
Absolutely. Yes. Yeah. No, it's part of our ethics and it's part of the workflow. We have no idea the patient that these come from. We just grow them and treat them as a patient almost, but we don't know where they come from. Yes.
so when we think about the feedback loop, so if we get to the stage where we can understand that these particular lesions respond well to one type of treatment or modality and others, how do we then have that information fed back to the patient
So
eventually?
yeah, so that is the hope, and that is what we are trying to develop, is that this becomes essentially like a pathology service. So you go to your doctor, they take a sample, they send it to the lab. in 2 weeks' time, we come back to you and say, this is the drug that's going to work for you. It's a lot of work to do that, but essentially that's what we're hoping is that we can personalize it for you. We can say, you know, like a blood test, we know what you need from this information. So yes.
It's so cool. Let's get through some of these questions. If people do want to stay up to date with the work on IMB, you can go to IMB endometriosis, just do a Google, and there's a spot that you can pop your email address in. We have been a partner of IMB for a really, really long time, and we share every opportunity that's going on as well. There's lots of initiatives like open days or come and check out the lab days that IMB advancements do. So yeah, hop onto their website to register your interest, or you can always stay up to date with us. So just going through some of the questions here, and Brett, you're very welcome to take them offline, and we can even throw them to some of the clinicians to respond to if it's not your wheelhouse. One of the questions was, does this work in conjunction with other medications? So this individual is on Ryeco for adenomyosis and wondering if it would help deep infiltrating endo, even that they were on Ryeco.
So Rayako, I believe, is a GnRH analog. So it's very much in that same class of drugs that we're already talking about. And it is just a slightly different preparation from say Dienogest or one of these other ones. And so I guess that's the question is I can't give you an answer to that directly, but I think what works for one person might not work for the other person. And so I don't know what is going to work for the individual. We need that to be tested to know for sure, unfortunately.
Mm. I think I'm just looking at my notes. I think it's a GnARH antagonist. Ryeco
Yes,
is.
yeah.
Yeah,
And I believe
not
it has the estrogen add back, so there's a little bit of estrogen in there as well to avoid the side effects of estrogen.
Yes, yes, they've got a
Ovulation.
couple in there. Someone has asked if they could take Digest, I can't ever get that word right, in menopause and in with HRT. Do you know?
Um, so firstly, I'm not a clinician. I can't answer any of that specifically, just to be aware. Um, and yeah, I think that's probably a best question for
Yeah,
your doctor, not for me.
no problem. Take that offline, chat back to your health professional. It's a good one to ask the question with them. Um, so um do people who are utilizing IVF increase inflammation that you've seen from any research?
Uh, None from what I've seen, and I guess the question for that is the location of the inflammation that you're talking about. Um, if you have endometriosis and are going through that IVF process, that's I guess a different question to whether or not if you're just talking about inflammation in general and IVF. Uh, I think I can't give you a definitive answer for that, but what we generally tend to see is there is a suppression of inflammation from this type of drugs. So, yeah, I mean, again, there's a lot of caveats of the type of drug you're using and the situation you're in, but
Mhm.
it wouldn't be a specific problem, I don't think.
What do you see? If you had all the power in the world, all the money in the world, what would you like to achieve in the next 3 years?
I think in the next 3 years, we're on the cusp of, I think, getting to a point of where we've actually got something new to test that we have sufficient data that could go towards a clinical trial would be fantastic. It still keeps coming back to all of the work we see is that we're still looking at hormone treatments for endometriosis. That hasn't changed for 30 years. Even a lot of the data I was showing before is still looking at glucocorticoids, which is a hormone. Uh, if we could get something that is not hormone related and is stratified to a specific patient population, I think that is something that would be a stepping stone to something a lot bigger. I'd like to see more than that, but realistic in 3 years, I think that would be a great goal.
Yeah. But I don't want you to be realistic. I want you to really, sky's the limit. Tell me something that you really wish if we could push for things, what would the your all in the power of the world in research, what would it look like?
Well, I think that from the basics, if we could really get a good hold on on what are the subtypes we're dealing with. If
Yeah.
we know,
Talk me through that. What do you mean by that? Really break it down because I think that's a really key point and it's not well, not a lot of people know about that. So talk to it through us what you mean.
yeah, I guess from the start, we have a really significant amount of presentation of endometriosis patients. You know, no two patients come in with exactly the same symptoms. So from that point of view, we're already dealing with a big amounts of differences. There is patients that have uh, pain, nociceptive pain that may not be driven by the lesions anymore. It's something that may be set up centrally.
What does that
There,
mean? What does nociceptive pain mean?
so this is the sort of pain when you have your nervous system fires continually. You have and when I say fires continuously, that's because it's being stimulated all the time. So if you're getting a nerve ending being activated constantly in your constant pain, your nerve changes. It adapts to that and sets up basically a lower threshold to be activated, unfortunately. And so your that peripheral nerve, that the one that's getting that activation, is changing what's happening centrally, and you have a setup where you have pain. You now have a different interpretation of pain, and so irrespective of any disease modification, you may just be dealing with pain that is there all the time. And there's different types of pain, and this is essentially what we call a central sensitization of pain. And so that creates a real issue for treatment, is that modifying the disease itself, removing the lesions, isn't necessarily going to have any sort of impact if you have this central sensitization. So that is one aspect of the whole thing. Just from the start, we need to understand if the pain is coming from something that has already been set up centrally, or is it something that we could be modifying at the patient at the lesion level. So already we've got a level of complexity where it's getting very difficult to research this. It's getting very difficult to actually treat this as well. So that level aside, then we come down to the fact that lesions can grow anywhere. They can grow on top of the bladder. They can grow on top of the bowel, on appendix, on ovaries. So they're growing in different places all the time. So whatever sets up those lesions, even if that is a similar situation, they're likely to progress in a completely different manner as well. A lesion that's growing on a bladder probably has different influences on it than a lesion that's growing on the bowel. And so then again, we need to understand what is the variations in all of that before we start even looking at modifying those lesions as well. And so all of that sort of feeds into a really complex state of almost a collection of diseases rather than one individual disease. And so it's going to always be really difficult to get an answer to anything if we're trying to lump in something that has been central sensitized and you have pain that is being created because of changes in your nervous system, to focusing on a lesion that is influencing something in the bowel, to something that is growing on the appendix. So there is so many levels of this that we need to really get to before we can start getting clear results in our research and clear results for treatment as well. So yeah, when we talk about getting to a level of understanding the disease, if we can at least start grouping those into smaller groups and knowing how to study and how to treat those smaller groups, that's going to be a huge benefit to moving forward as well.
I love this stuff. So thank you, thank you, thank you for doing that work. It's just so these are missing pieces. And they have been for a long time.
Yes, yeah. I think it's very easy to, like, from a research point of view, sometimes we just lump it in together because that makes it easier and makes the sample numbers higher, and we can just do it this way. But we lose the subtlety in all of that, and we lose these differences. And one thing that I always think is really critical is that we know that there's a diagnosis delay from 7 to 10 years. So when we see a lesion, are we seeing a lesion that's 6 months old? Are we seeing one that's 2 years old? Are we seeing one that's 10 years old? How do we know if the treatment of one that's 6 years old is going to be the same as one that's 2 years old? And I don't, it's a question we think about in the lab all the time. How do we identify that?
You don't have a
I don't know.
benchmark.
Yeah,
Yeah.
yeah, it's when we don't know when it starts, so we can't say how long it's been there.
And it's not ethical to just send people for laps and
No,
you never get that part with ethics committee just to
no, no, and even then, you still don't know when it starts. It's
mm,
like there's no marker for this. So,
yes, yes. Someone has asked if could be possible multiple causes or triggers for the endo lesions.
absolutely, and it's a theory that we sort of work under. Actually, is that part of this variation in the disease is the fact that it's started by many different reasons. We know that there is a genetic basis for endo. Uh, what exactly those genetic variants do to create the endometriotic lesion is not clear, but we kind of believe that there is groupings of that. So if you get, so I think at the moment there's been about 45 genes that have been associated with endometriosis. Say if, and you can have all 45 of those variants and still not get endo. It's a complex genetic disease, so it's not like a variant equals disease. It's a complex mix. But we kind of think of that theory, maybe you have 20 of these ones and they all hit the same pathway and they're all affecting the same thing. So you end up with a lesion that develops of that type. Whereas you have another 20 that are all affecting maybe the way the inflammation is working in the body. So you get a lesion that is more inflammatory stimulative or something like that. Now, this is very speculative. This is just theories, but I definitely do believe that there is multiple causes of endo. And that once we get to that, we'll start to get to that idea of stratification as well.
Um, what about epigenetics? Can you touch on that? Your thoughts?
Yeah, I think it's the same concept. There is a contribution of epigenetics to the development of lesions, but it's each one of those is small and accumulative. And so
Mm.
it's the way that they accumulate together is going to increase susceptibility, but not necessarily be prescriptive of the disease. And so I do think it's the way they accumulate and the combinations that may underlie the types of diseases that will eventually manifest as well.
And the role of estrogen, do they feed the lesions per se? Is that a trigger for them to grow?
Yes, very much so. So we consider it an estrogen dependent disease. It's part of the reason why the current drugs are oral contraceptives and progestins is they reduce systemic estrogen. So there is a downregulation of systemic estrogen. Systemic, I mean in the bloodstream through the whole system. And so that should stop the lesions from growing. Now, again, come back to the fact that not every lesion is the same. That doesn't work for everybody. There is also evidence that lesions can start producing their own supply of estrogen as well. So even when you are suppressing systemically, the lesion may be getting the estrogen as well. So yes, the estrogen does feed the lesions, and it's one of our most currently successful way to try and stop them growing.
Thank you so much, Brett. Thank you for everyone for your questions. Brett, do you have any questions for the consumers or the health professionals on the line here at the moment? Anything running around your mind?
Um, I have lots of questions. I don't know how much time we've had, but I do think it's critical for us to know what are the biggest problems. What do people need answered for this? Only then can we really design our best research questions. So I would like to know what it is that people really want to get answered. I'm not sure we can do that in just 1 or 2 minutes, but that would be my question. What is the major problems? What do you feel we need to solve?
Beautiful. People can email us info@quendo.org.au, and we're always collecting questions. We have a research question library that when we meet with the team at IMB and others, or when they propose an idea to us, or vice versa, we can see what is the interest in the wider community. So, as Brett said, the sector, the wider group, you listening, actually needs to inform the research, because at the end of the day, this is taxpayers' money, the MRFF and NHMRC and other donors, etc. So it really is a collaborative, and we absolutely love working with IMB and what Brett's doing, and Dr. Akwazi Akamoto and so many other people in that group. So yeah, Brett, thank you for flying the flag still and being curious still, even though sometimes, we just want to bang our heads up against the wall. We really appreciate it. And if anyone has any, messages of thanks for the wonderful researchers, please feel free to share them now or email us and we can forward them too. It's been such a wonderful evening, and yeah, it's so cool to see this type of work. It is like nothing before, and as you say, I feel that it will really help move the goalposts as well in terms of what are we striving to next.
Yeah, yeah.
Great.
Thank you very much, Jess.
Thanks, Brett. Love your work. We'll keep chatting. Like I said, if you want to know more about this, please reach out. We have consumer advisory groups going all the time, and specifically on the ones that Brett has mentioned. So please email us and we can send you the correct information and go from there. Have an awesome night, everyone. And yeah, take care. Thanks again.
Thank you.
Thanks, Brett. See ya.